Louis Martin's going to talk to us about a different way of detection of TB and possibly some effects for all of us, because this is another one of these things that hasn't seemed to have gone away on us either. So with that, I'll turn the mic over to Louis.
Shorter they are.
I don't feel offended that most people tell me that.
First of all, I'd like to thank Harvey and Harley and the Alliance for the invitation to talk about my new area of interest these days. It's always a pleasure to be here with this group. You're a very active group, very knowledgeable, and it's always good to talk about what's going on from my perspective as well. Now, as Todd mentioned, or Doug mentioned, it is getting into siesta time, so rest assured I had a couple cups of coffee, so I should stay awake for my talk. Well, and I guess I'm one of the few wearing a tie, so I should explain this is from my early training as a scientist. I found ended up working in laboratories where it's a mix of science folks and a bunch of clinical fellows, and we tended to dress down in the lab, and if anybody showed up in a tie, if they were their clinician, it meant they were doing clinic and working sometime during the day. As a science guy, we were either going to a wedding or doing a presentation. So if there's no justice of the peace in the audience, I'll get on to the presentation side. I guess I should get my presentation.
That's Hawaii, by the way.
Yeah, I should talk about Hawaii. There's the others.
Try the mouse.
There we go.
Okay, so I'm going to talk about a diagnostic technology that's coming out of the University of Nottingham in the UK, and a new company set up there, PBD Biotech, that's trying to commercialize the technology. When we're talking about TB, it's caused by a bug or mycobacteria, which is related to the other gram-positive bacteria that we come to know and love, such as Listeria, Staphylococcus, and lactic acid bacteria. Gram-positive means that they have a thick cell wall that stains with this gram dye, which is a nice trick for microbiologists to characterize things. What it means to us is that it's tough to get at the organism because the cell wall is quite strong, durable, it's resistant to breakage, and it's resistant to attack as well. And the other bit that gives us a problem is that these pathogens tend to grow rather slowly in culture. So if we try to take an isolate out and put it into culture and grow it, we're waiting for 1 or 2 months to get the data out to identify what the heck we have going on. And this second slide is probably the most important slide in the presentation. I'm going to, for those of you that haven't yet got your PhD in immunology, I'll spend a bit of time on this because it is a key for diagnostics and will apply to TB or anything else you're trying to diagnose. If I can compare it a little bit to my previous vaccine world, there's a lot of stuff, a lot of complications and things that Scott Napper went into some of the aspects that go into the black box to make a vaccine. And as an end user, you really don't care about that. All you need to know is it effective and how many times they have to inject, and if it's more than one injection, how far apart do they have to be. For diagnostics, it's a little more complicated because depending on what you're diagnosing, what you're detecting, you're going to get different readouts and different levels of efficiency. If you will, if you're walking around in your normal day, you detect, you diagnose your environment using your 5 senses. For some things you'll hear, for some things you'll taste, feel, or smell, or enjoy your lunch with using several senses at the same time. the same for diagnosis, is you have to know what you're detecting. Because different detection methods will pick up different parts of the infection and will give you different readouts and at different times and different efficiencies. So in the course of an infection, as you have the initial stages, you'll have the bug starting to grow, and there really isn't much of a response to pick up on. Normally we'll look for antibodies or we'll look for some signaling molecules, but as the infection grows, then we get an antibody response, so things like ELISAs or immunoassays pick that up. You can also get signaling molecules. Gamma interferon is, if you will, a hormone of the immune response. It is a signaling molecule in the immune system that triggers immune cells to react to an infection. And again, as the infection grows, that goes up. So you can try to measure with a tuberculin skin test and TB for antibody responses. You can measure for gamma interferon and things like that. The problem is that those will happen after the infection is taken, and so it won't pick up early infections. And the other downside is if you vaccinate your animal, you will get antibody responses. You'll get positive tuberculin skin test. So if you vaccinate, you can no longer diagnose on those assays. So you have a conundrum there. Do you want to vaccinate and protect, or do you want to not vaccinate and detect? You can't do both. And that's where the Actiphage, our kit, comes in, is we're trying to pick up the actual organisms at the early stages and anywhere through the infection. So what we do in the Actiphage is we look to identify living mycobacteria, so the organism that's causing the infection, the TB in this case. And what the bacteriophage are really organisms that attack mycobacteria. So you can, so that's a phage particle there. It's quite distinctive looking, and it will bind to the surface of the mycobacteria and injects genetic material that reproduces inside the bacterial cell, and eventually after reproduction it will change its life cycle to a lytic phase, and it will break open the cell, releasing genetic materials. And the mycobacterial genetics will just be put out there, no longer living, and the phage will go on and infect another bacteria and keep repeating the cycle. So we can take advantage of that. Sorry, that's just this is a better photograph showing a lysed mycobacterium, and these are infected that are still getting phage replication but haven't yet got to the lytic phage. So it's a nice mixture, and this allows you to one, to get lysed material, so you're no longer worried about the difficult to disrupt the cell wall, because the phage have done that for you. And by identifying these, you're able to then obtain genetic material from these healthy ones around and identify what the specific bug is. Our initial assay did this in entirety. We were able to plate out the blood sample and get these lytic colonies here. Each of these represents probably one parental mycobacteria that has grown and then been lysed in a population. And so you're able to go in there with a fine needle, pull a sample out, and then do DNA sequencing to identify the identity and the infective agent. This type of assay will take about 2 days to run. And essentially, schematically, this was it. We would take your sample that you had from your animal, add our phage to it, let it internalize to live mycobacterium, not the dead ones. It only goes into living. Then you clean up the outside to get rid of any non-incorporated phage. Let them grow for a little while, culture, let that go for a day or so, pick the clones, and then you're able to look at the DNA and see what you've got. And it's definitely identifies the bug. It's not necessarily quantitative. You get some you have to do some other tricks to do quantitation, but it really does give you an infection or no infection readout. This was done in the UK in a couple of studies. We were able to, and they were published. It worked well to pick up Johne's disease, which is TB. And we also published on TB itself. This was done with the original 2-day assay, which was good, but we wanted to get it faster. So there's moved on to a totally liquid assay where we don't need plating. It's done in the tube. We take the material, we purify the peripheral blood mononuclear cells, essentially the white blood cells, from the blood sample, and then those are mixed with the phage. Let that incubate for about 3-4 hours, and they go straight to the PCR assay, which is a DNA amplification assay, which lets us amplify the number of copies of the gene that are present using specific sequences. And then we're able to detect less than 10 mycobacterial cells in a ml of blood. In some cases, we've gone as far down to just two cells per ml of blood being detected. So we have very nice limited detection in the assay. And by using the proper DNA sequences in the PCR reaction, we're able to keep the reaction specific to the species of mycobacterium that we're looking for, be it TB or para-TB or something else. Okay. We did in the early studies, we did do a comparison of the actiphage assay with the other standard assays there. Did quite well. Definitely much more sensitive with gamma, which I think now is starting. It was being used in the U.S. quite extensively for TB testing in cattle, and that has ceased over the past year. It's really not a very sensitive assay. The skin assay still remains the mainly used one, and we matched up well with that assay, and in fact we're able to, working in conjunction with the skin assay, provide much better specificity to it. And of interest, I think, to this group particularly is beyond the cattle and sheep, goat studies that were the initial foundations for the work. We've now been working, particularly in the UK, with groups in the zoos there to check out into other species. So the assay is working for species beyond the traditional farm livestock. I believe the list now is up to about 20 different species that they've identified mycobacterium TB in, working with some zoos in the UK. And hopefully over the next year or so working here, we'll add cervids to the list. There's more there. The work so far, and let's acknowledge the people working on it, as you see they're in the UK based. They said this is originated in Nottingham. I started with the company about 2 months ago now. We're trying to set up a working group in Canada based in Saskatchewan, initially working with the Vito group, who's got some of our kits now starting to functionalize it here. We've also introduced it in the US, a couple of labs in Colorado and Pennsylvania are working on it in cattle. So we're hopeful that over the next year there'll be a very rapid expansion and growth of using the kit and having it available in Canada. Excuse me. And building out for other animals beyond just the traditional cattle. So I think that's it for now. If you have any questions, I'll be happy to answer them.
First question, answer may be obvious, but I want to hear it anyway, is you are obviously detecting live bacteria rather than antibodies to that bacteria. So you're not testing for exposure to, you're testing to actual disease presence.
That's correct. That has to be an active infection.
Good, okay, thank you. And what kind of a reception have you got from CFIA towards this test?
They're extremely interested in it. They're extremely picky that we get in for permits before we bring it in. And we absolutely do no testing for TB without their approval. But I think overall it's very positive. They want to see the data. They want to get some kits in their own hands to run it as well.
Thank you.
More questions? Oh, to the back.
What is the difference between your test and the current test being used in the States?
Which test in the US are you referring to?
Like they use a blood test, a blood TB test in the States for cervids?
Good.
I think that's the ChemBio test,
Is that what?
DPP test. It's
I'm
an
not-
antibody-based test.
Yes, I think that's what I was going to.
So similar to what Ian said.
It's an antibody ELISA-based test, and it's about 50% accurate, so.
This test, is there any way to decipher, like when you do have a positive, do you know if it's an avian positive or a bovine positive?
I guess we can do the specific primers to differentiate between the two.
Okay. And do you know cost per animal?
I've asked that question, I haven't got a straight answer yet,
Okay.
but it will, we're still trying to determine what our business model will be. Where it ties have to talk business. So there'll be kits for sale, so they could go to wherever. But we're also trying to establish our own laboratory, so samples can come to us and can run, so there'll probably be a different cost structure between the two.
Thanks.
Nick?
Can you just repeat what the accuracy of the TB test in the US was that's been approved?
I think the ELISA test is about 50%.
Okay.
So flip a coin.
Okay. I'm just saying, we're arguing about a CWD test that has 80% potential sensitivity,
Yeah.
right?
I think still the skin test is the best one that's still available. But unfortunately, that's an immune response, which may not be reflective of an active infection anymore.
Questions from the back?
In the States, in our TB zone in Michigan, we had a hard time determining between animals that are actually infected with TB and animals that had been vaccinated with the TB vaccine. Would this test be able to tell the difference between them?
Exactly, that's one of the points that we particularly like about this test, because it's independent of antibody response. So you could, if you want to vaccinate your herd and still be able to run the diagnostic, you could get an accurate assessment of whether you have any infection or not. Yeah. Would I just, full disclosure, if there is a downside to our test, is that there always when you're trying to detect the bugs themselves, there is no amplification, so there will be a limited detection, be it 2 or 10 mycobacterium per mil of blood. If you're below that number, you'll get a false negative. You won't be able to pick it up. So if you're at a very early stage of infection, or a lot of infections go up and then they come down, and they go up and then they take off, if you happen to sample when they're in one of those low valleys, you might miss it. So no test is ever going to be 100%, but confident we're be in the high 90s with this one.
Further questions? Going once? Going twice? Done.
Not a question, but more of a comment. Just, if you are trying to put this towards CFIA, I think one of the beneficial things you could look at is just the cost that CFIA incurs. And potentially having more people, I mean, like a lot of the producers in the province, many of them aren't TB testing because they don't want to deal with the hassle of it. And running animals in twice. And I mean, CFIA has to go out for 2 days, 2 different days to a farm to do this. And they have to, you know, I don't know what their lab costs are, but if animals are injured, those animals, they have to pay compensation for that. So if you cut the handling time in half, then you're reducing that cost to CFIA. And you'd also see probably an influx of people TB testing that were not TB testing before, because if it's a one-shot deal, then more people will be willing to do it.
Okay. Well, definitely, this will be a one-shot deal. We take one blood sample and we're done. Now, if it's negative because of the reasons that you might have missed a negative, if there's reason to suspect that the herd had been infected at some point, you may have to come back a year later and do a second test and get 2 negatives before you can say the herd's clear and don't test anymore. A lot of that's going to be a CFIA decision on how they want to keep their surveillance going. And as you say, I think that the prices can be complicated. Like the people investing in the company want $1,000 a test, and that's not going to fly. And so it's going to depend on the cost-benefit analysis. So we still, I think, are working through what the pricing is going to be.
High throughput you had on one slide there, what kind of throughput are you talking about? How many samples in an hour or whatever?
Manually, we can run about 50 samples in a day with one person, primarily the blood separation step that's slow. But I think that's something we're going to be evolving to run through more. Right now, we're looking in the Colorado lab to run, I believe, 500 to 1,000 head of cattle through in less than a week. That's still the manual method.
Yeah, they're not hearing it back, so. No. Any further questions? Okay, if you missed them, well, like you said, you're here for the panel afterwards. Okay.
Yep, I'll be here until about 4 o'clock.
Okay.
Thank you.